Hello everyone, this is Chinmoy from Chinmoy's Biology channel. So let's get started with a very important topic, linkage and recombination. So please do pay attention to this full episode because this is a very interesting and a quite difficult topic. So let's get started. So what is linkage? So linkage is a phenomena of certain genes which are staying together during inheritance through generations without change or separation as they are present on the same chromosome. So what is that you can see here? These are the chromosomes and these genes are located on this chromosomes. Okay, so we know that genes are located in a specific locus of a chromosome, and if these are located like this, they are linked. So more they are closely located. If the genes are located in this part of the chromosome, the linkage will be more. So the linkage here will be greater. But if one is located in this part of the chromosome and one is located in this part of the chromosome, the linkage bond will be less. Okay, so the linkage strength will be less. Like this they are located. So suppose this is A, B and this is A and B. So this is a very strong linkage. So these two pairs would not be able to broken. So they will move as this whole part only. But this you can break because the linkage is less tight. Okay. So that is this is what they are inherited throughout the generations without change on separation as they are present on the same chromosome. So if they are present on the same chromosome and they are closely related to near to each other, they are inseparable. So without changing their location, they are inseparable. They move on from one generation to another. So let us take an example. Suppose you all four of your friends are very close. So you sit on the same bench, you go for a movie together, you go for a picnic together. So you are inseparable. So if one of your friends are going to one movie, if you are not liking that movie also, three of you will move with that friend because you are linked, you are having close bonding to each other. So if suppose if you are going to study, suppose one of your friend is going to study maths, if you are not feeling to do maths at that time, you want to do chemistry or biology, but as he or she is doing maths, you will have to do that because you are having a good bondage. So this is the linkage which is telling that they are so closely linked that they are inherited through generations. They are inherited through generations without separating as they are present on the same chromosome. So same as your friendship, you are closely related, you are very close friends, you will move on to from one place to another together only. So this is an example. So who first proposed this? This was first proposed by William Patterson and Punnett, R.C. Punnett. They discovered actually this linkage, but they couldn't do much more experiment to prove it. It was formulated, the concept of linkage was worked by Thomas Morgan and his works on Drosophila melanogaster. Okay, so on this Drosophila melanogasters or fruit flies, he worked for years and then he proved this linkage and recombination. So regarding this, why he chose Drosophila melanogaster, that we will read in a separate video in a separate class. Today we will just focus on linkage, recombination, crossing over and all. So stay tuned throughout the full episode. Don't skip the episode in between. If you are skipping, you will miss the full important episode. So stay tuned and please do subscribe because to know more regarding this principles of genetic, molecular basis of inheritance, there are many more interesting topics which are coming in the next videos. So now let's move on to the next part. So here we can see that linked versus unlinked genes, how they look like. So if here these are unlinked genes located on different chromosomes. If suppose in here is a gene 1 and in another gene in another chromosome, sorry, there is a location of this gene over here. So if these two you are taking, this 1 and 2, these genes are unlinked. But here you can see gene 2, gene 3 and gene 4, they are on the same chromosome and they are located very close to each other. So this is a linkage group. So a set of genes at different loci on the same gene that except for crossing over tend to be inherited together. So except crossing over is happening, that this part is being crossed with another part, until and unless that is happening, crossing over is happening, this whole gene part will be inherited together. Okay, so we can't break this, you can't inherit this genes separately. So if you think that gene 2 will be going to one chromosome, gene 3 will go to another, or gene 4 will go to another chromosome, it's not like that. They will go in a group. So this is a linkage group. So this is a linkage group, they all will be inherited together in the next chromosome also. But if crossing over is happening, then they will be inherited uh, separately. You can do this in crossing over. So what is crossing over? We will know that now. So what is crossing over? So crossing over is the exchange of genetic material between the non-sister chromatids of homologous chromosomes during meiosis. So first let's discuss what is non-sister chromatids and what is sister chromatids. So these two, let me draw in different colors. When I'm taking these two with the same green color, these two are the sister chromatids. Okay, these two are the sister chromatids. Now if I'm drawing another chromosome like this, so one of this and one of this, these are non-sister chromatids. Is it clear to all of you? So again in this two are sister chromatids, that is which are paired together. So these are again sister chromatids, and this one are non-sister chromatids. So the genetic material exchange will happen between this one and this one, between only the non-sister chromatids during meiosis, which results in the new combinations of daughter cells. So the daughter cells which will be formed will have a new combination having a part of this gene, genetic material from this chromatid, and a genetic material from this chromatid. So daughter cell will be a recombination of both maternal as well as paternal chromatid. So that is a recombination. After crossing over happens, a recombinant new cell will be formed. So here in the diagram you just see what is happening. So pairing of two homologous chromosomes we are seeing here. This is paternal coming from the father and this is maternal. So crossing over is happening here. You can see the genes A, small a, capital A. These two are small a in this orange part and these two are capital A in this brown or deep red chromatids or chromosomes, whatever you are telling. Now crossing over is happening. So one section of genetic material of this orange has been cut and separated and forming this part, and another part has come and joined this part. So now what are the new chromosomes looking like? That is the daughter cells. So they are having this one strand is perfectly okay, same as parental, but the next part is having a recombinant part. So one part of the parent and another genetic material has come from the non-sister chromatid. Is it clear? Now that is the capital A. It has come from the non-sister chromatid. The same has happened in this part also. Here you can see in this deep red color, one part of orange has mixed. So this is also a recombinant part. That part was having a specific gene. So together with the gene, the genetic material has been crossed over to those non-sister chromatids. So this is crossing over. I think it is clear to you all. If you are having still any doubts, please do write in my comment box, or I'll get back to you with another episode of this lesson. And please rewind this episode again, go back from the beginning, rewind and go through it. So is it clear? Now let us move on to the next part. Now here crossing over, another diagram is here. Now just let us see. Here you can see this A and this A. This is a capital A, this is small a, and here B, C genes are located here. Capital B, capital C, small b, small c. So first the recombination is happening like this. You can see this full this red part is as it is, and another section of this blue part has gone here, and this red part has cut and gone here. So this is mainly, we can see that crossing over of the region of A B recombination. So A as well as B, both the parts recombination has happened here. So upper part, that is the chromosomes, so the non-sister chromatids A region, also genetic material has been crossed over, as well as the B region also. Now in the second crossing over, what we see here, that part is intact, A region is intact as it was in the previous crossing over, but the BC region here, this part has gone here and this part has gone here. So that is a BC recombination. So this is what keeps on happening generation after generation. But these recombinations or crossing overs are very rare. Generally, we move on from the parental, the inheritance of characters is mostly we can say it as a normal type phenomena. Crossing over and recombination are generally very less. So that also in the diagram we will see, as per the NCERT book, that full NCERT full diagram we will discuss in the next class itself. So that is a very important diagram, but in your book NCERT it has not been explained properly. I'll take the diagram only in the next class and I'll discuss the diagram full in details, and how easy it is. Once you understand that diagram, it will be very easy for you to understand linkage. So now we will see what is recombination. So what is recombination? So genetic recombinations occurs when genetic material is exchanged between, we can say that when genetic material is exchanged between two different chromosomes. Okay, it is exchanged between two different chromosomes and between two different regions within the same chromosome. We can observe it in eukaryotes, that is like animals and plants, and prokaryotes, that is an archaea and bacterium. Now we can say that we should keep in mind that in order to exchange an exchange which is occurring, the sequences containing the swapped regions have to be homologous. Okay, they are not happening in heterogeneous chromosomes. Okay, so they are always happening in homologous chromosomes or similar type of chromosome, or to some same degree. Okay, so we will tell that in heterogeneous chromosomes that these recombination will not happen. So the genetic material is exchanged between two different chromosomes, but they should be homologous. So let us see here what is happening. So these two are homologous chromosomes. These are non-sister chromatids and these are sister chromatids. I have told you earlier. So this is bivalent. So during this is we can say during meiosis, this chiasma is formed, and this part has been come to this region. This has come to this region. Again, this genetic material has gone to this, and this has gone to this. So these are what? These are the recombinant chromatids. So this is how the recombination has occurred. So this process is very important, and this is recombination. So some part of genetic material from the this parent has gone to this part, and some has gone from this part to this part. So there a new combination of chromosome has occurred, that is a recombinant one, that is having recombinant chromatids. So if you understand this clearly, then only you will be understanding the recombinant DNA technology in the higher classes or in long term. So just make it clear, these parts of genetic material from one chromosome to another chromosome are being shifted, are being exchanged. So they are not moving anywhere else. They are just taking from one and moving to another one. They are exchanging. Is it clear? So this will help us to learn the recombinant DNA technology. Okay, so we will also learn the molecular basis and all in the next classes. So let's move on to what is the difference between recombination and crossing over. So this is the last part of recombination and linkage. So recombination and crossing over, what are they? So recombination and crossing over related, but not exactly the same, because recombination is the production of different combinations of offspring. So after crossing over, this recombination is coming when different varieties of offsprings are produced which are different from the parent, And after that, genetic material from the parental to chromatids has been passed on, that is from the non-sister chromatids, this exchange has happened. So that exchange, we call it crossing over. So what is crossing over? Crossing over is the exchange of genetic material between the non-sister chromatids during the meiosis, which results in recombination. Okay, so when this non-sister chromatids are passing on their genetic material to the exchanging the genetic material, in that cases only in the next generation the recombination will be happening. So this is a difference between crossing over and recombination. So this is all for today's episode. In the next class we will be studying with a few more things of linkage, and firstly in the next class we will study about why Thomas Morgan took drosophila as his experimental animal, and what are the benefits of that, and why he took those for his experiment. So that will be the next class, and after that we will come to again your diagram in NCERT book, that in details I'll teach you. So stay tuned, subscribe to my channel so that you are not missing all my updates, all my videos. Thank you. Thanks a lot for watching.